Hello everyone. This is part 2 of our RNA splicing lecture. So today we will talk about how does splicing take place. Before we proceed towards the mechanism, how does splicing take place, we need to understand the basic composition of an intron and what are the different sequences that are needed for the splicing to take place. Here you see a pre-mRNA molecule. This is the 5' exon. This is one exon, and this is second exon at the 3' end. So in between these two exons, we have an intron present. So we have one exon at the 5' end and one at the 3' end, and this is the intron in the middle. We will be specifically talking about the consensus sequences of the splice sites. So in order for the splicing to take place, an intron needs to have 30 to 40 nucleotides at each end of the intron with specific sequences. So this cut in the middle means there's a long range of nucleotides in the middle, and we need to look at two consensus sequences, the consensus sequence at the 3' splice site, at the 5' splice site, and the consensus sequence at the 3' splice site. So what are these sequences, and what is the function of these different sequences? So there are some invariant bases both at the 5' end and at the 3' end. Invariant bases means they do not change. You would always, for example, find a GU at the 5' end and an AG sequence at the 3' end. As you can see, the number here indicates the frequency of occurrence in any intron. So 100 means almost all the introns they would contain a G at their 5' splice site followed by a U all the time. And the same is true for the 3' splice site, that an intron at its 3' end would contain an A and a G 100% of the time, so all the time. The other numbers like 80 or 60 indicates that 80% of the times you would find a C here. And so 100% of the times A. This is another invariant. So 80%, 90%, 70%. So that's the frequency of occurrence of different nucleotides in an intron. But here we have these two invariants, one at the 5' site, that is the GU, and the 3' splice site contains an AG invariant. These are invariant bases, means they do not vary, they do not change. You would always find a GU at the 5' end of the intron and an AG at the 3' end of an intron. Another invariant here is the branch point. This is always an A, an adenine, and this adenine or this branch point is generally 20 to 50 bases upstream from the 3' end. And upstream from the 3' end, we also have a pyrimidine rich region, which is roughly 15 bases in length. We will talk about the function of these different regions later, but the presence of the GU and AG site at both ends of the intron and this branch point in the middle and the pyrimidine rich region, all of them help in the process of RNA splicing, where the intron is removed and the two exons, the one at the 5' end and the one at the 3' end, they are joined together. So in order to remove this intron, we would always need these sequences, the 5' GU region, a GU sequence, 3' AG sequence, and a branch point sequence. So they're fixed, they are invariant bases. We have previously discussed that there are certain factors, U1, U2, U4, 5, and 6. If I can go back to the previous slides. So we have these 5 SNPs which are involved in splicing, U1, U2, U4, 5, and 6. And we have also discussed that these SNRNPs, they are composed of two things, a protein and an snRNA molecule. And I told you that this snRNA molecule, these molecules contain particular sequences here, which can recognize a sequence at the intron, and they can base pair with it. This helps these snRNPs bind to the RNA molecule. So how do they recognize this sequence and how do they bind to this sequence is that is shown on this slide here. So this is again the same mRNA molecule. We have this exon 1 at the 5' end, we have exon 2 at the 3' end, and in the middle we have got this intron. And as you can see, this is a GU at the 5' end and contains an AG at the 3' end. These are invariant base pairs, and it also contains an A in the middle, an adenine. This is the pyrimidine rich region, and this is the branch point. This is a pre-mRNA molecule, and two snRNPs, U1 snRNP and U2 snRNP. U1 and U2, they initially bind to the primary mRNA molecule. So what is shown here is not the protein, but just the snRNA molecule, so that we can better understand the base pairing between the mRNA molecule and the snRNA molecule. Just imagine that this snRNA molecule is associated with a protein here. So there's a larger protein here, and this is the RNA molecule of that protein. So together they constitute snRNP. So U1 binds at the 5' end of the intron, while U2 binds to the 3' end of the intron. As you can see, that the U1 snRNA has a complementary sequence to the 5' end of the mRNA, which overlaps with part of the exon as well. And this complementary sequence helps the snRNA molecule recognize the 5' end of the intron and bind to it. If we look at snRNA2, U2 snRNA, this also contains a specific complementary sequence which binds to the 3' end, and it recognizes the 3' end of the pre-mRNA molecule and then binds to it. But what we see here, what is interesting here, that this branch point is bulged out. Why is it bulged out? This was an adenine, so the sequence on the pre-mRNA was U, A, C, U, A, and C. This adenine got bulged out because the complementary sequence of the snRNA did not have a complementary base pair, a complementary nucleotide for base pairing. So for U it contained an A, for A it contained a U, C, G, U, A, U, and if this A was inside here, the snRNA was supposed to contain a U. But because it does not contain a U, it rather contains a G, so G is going to pair with a C, and this kicks out this adenine. So this creates a branch point. That's why we call it a branch point. So this is bulged out by creating a branch here. And this is pyrimidine rich region where an auxiliary factor, AUF2, has to bind. So these three things initially need to bind to a primary transcript, U1 at the 5' end and U2 at the 3' end, and then an auxiliary factor here. And this creates the initial spliceosome or the primary spliceosome. Let's look at the mechanism of splicing. So the pre-spliceosome, as discussed previously, this is composed of this. We have the primary. We have this pre-mRNA molecule. This is exon 1 and this is exon 2, and in the middle we have got intron. And on the intron, this is our 5' end of the intron, and this is the 3' end of the intron. So at the 5' end, U1 is bound, and at the 3' end and the branch point, this is U2 is bound. And what we see here is U2AF. So U2AF means auxiliary factor, means this auxiliary factor basically binds to the pyrimidine rich region. It also contains two domains, a smaller one and a bigger one. So it binds to the 3' end of the intron and as well as to the pyrimidine rich region. And this basically helps U2 bind to the 3' end of the intron. That's why it is known as U2 auxiliary factor, because it helps U2 bind to the pre-mRNA molecule at the branch site. So they're bound at the 3' end and as well as at the branch site. So this constitutes a pre-spliceosome. We have got a U1, U2 and U2 auxiliary factor. So after binding of these two SNRNPs and this auxiliary factor, the spliceosome is formed by the addition of 3 other SNRNPs, which are U4, U6 and U5. So this combination basically then leads to the formation of a mature spliceosome. Where we have got U1 at the 5' end, U2 at the 3' end, and then we have got U4, U5 and U6 also bound to the pre-mRNA molecule. Once this mature spliceosome is formed, this creates the first catalytic complex, and this complex kicks out U1 and U2. U4. When U1 and U4 are kicked out, they reorganize themselves. U2, U5 and U6, they reorganize themselves and carry out their first catalytic activity, where the branch point attacks the phosphate group at the 3' end of the first exon. We will see this in details on the other slide. You will understand it better. But what you need to understand here now, that an initial spliceosome is formed by the binding of U1 at the 5' end and U2 at the 3' end, and then later U4, 5 and 6, they join the pre-spliceosome leading to the formation of a mature full spliceosome. This then kicks out U1 and U4, and the U2, U5 and U6 are reorganized, constituting the catalytic spliceosome, which would carry out this cleavage functions. So basically there are two cleavage functions. There are two cleavage steps which would take place here, two types of cleavage. One cleavage is at the 5' end and one is at the 3' end here, because we need to remove the intron. So we need to cleave it here at the 5' end of the intron and at the 3' of the intron. So the first cleavage takes place at the 5' end, and the second cleavage takes place at the 3' end. And how does this cleavage take place? This is first of all the cleavage at the 5' splice site. For this to take place, so this is what we call first transesterification reaction. So two transesterification reactions are needed, one at the 5' end and one at the 3' end of the intron. So in the first transesterification reaction, the hydroxyl group of the 2' hydroxyl of branch point adenine, because it was bulged out, so the hydroxyl at the 2' of this adenine basically attacks the phosphate at the 5' end of this of the intron. So this is the sequence of this is a phosphate group of the intron. So that's your intron, and this is the 5' phosphate of the intron. So the 2' branch point that attacks the 5' phosphate of the intron and cleaves it, makes a cut here, and after cutting it, it joins the 5' end of the intron with the 2' end of the adenine, thereby creating a 2' 5' phosphodiester linkage. So this is also a phosphodiester linkage. We have these two bonds here, one here and one here. So rather than a 5' 3', this is 5' 2' reaction. So this phosphate bond was cleaved here because of the attack by the 2' hydroxyl group of the branch point adenine. This attacks here, cleaves it here, and then joins the 5' phosphate with the 2' hydroxyl, leading to 2' 5' phosphodiester linkage. So that creates a loop-like structure going like this. So this creates a loop. It starts from here, goes like this, and then comes back here. Now 5' end has been dissociated. The 5' end of the intron has been dissociated from exon 1. We are left with a hydroxyl group at the 3' end of the exon. This is a hydroxyl group at the 3' end of exon 1. Now this, while this the phosphodiester bond at the 5' end of exon 2 is still intact. Now the 3' hydroxyl of exon 1 attacks the 5' phosphate of exon 2. So this is exon 2 and this is exon 1. So the hydroxyl group at the 3' end of exon 1, this attacks the phosphate group at the 5' end of exon 2, and it cleaves it, it cuts it off from here. And after cutting this off here, we are left with an excised lariat intron, where we have this 2', 5' phosphodiester linkage with the branch point, and here we have got a hydroxyl group because this hydrogen is added to the oxygen here, and we are left with a 3' hydroxyl group. So the intron that has been removed now, it contains a lariat structure, a hook-like, a loop-like structure, where we have this 2', 5' phosphodiester linkage, and this like a loop, and contains a 3' free-hanging 3' hydroxyl end. While here, the attack of hydroxyl group from exon 1 to the phosphate group on exon 2, this leads to the formation of a normal 3', 5' phosphodiester bond, or 5', 3' phosphodiester bond. So this is the 5' end and this is the 3' end. So a normal regular 5', 3' phosphodiester bond is established between the two exons, and that's how two exons are joined together. And the lariat intron has been removed. So if I repeat it, the first transesterification reaction takes place at the 5' end in order to cleave it off, which is carried out by the attack of hydroxyl group from 2' hydroxyl group from adenine, which was basically the branch point. It attacks the 5' end of the intron here and cleaves it off, and we are left with a 3' hydroxyl at exon 1. Now this 3' hydroxyl, this attacks in the second transesterification, this 3' hydroxyl attacks the 5' phosphate of exon 2, thereby cleaving it from here. This is the second transesterification reaction, and the two exons are joined together by a normal 5', 3' phosphodiester bond. So that's the 5', 3' phosphodiester bond, and this intron is then removed from this complex, and we are left with a lariat structure where we have 2', 5' phosphodiester linkage, and we have a free floating 3' hydroxyl end. What happens afterwards? Now we have got three things here. We have got the spliced exons, the exons that have been put together. This was exon 1 and this was exon 2, and the intron that has been removed from here. Here is the lariat intron, which is connected at the branch point by a 2', 5' phosphodiester linkage, and we have a 3' hydroxyl group which is hanging here. So this is the second product of our splicing reaction. And the third thing we have here is the catalytic SNPs, which are U2, U5 and U6. Now we need to destroy all of them except the spliced exons, our mRNA molecule. So in order to do this, because this has a lariat structure, this RNA molecule is protected. So in order to degrade it, first we need to debranch it. We need to break this branch here. This 2', 5' phosphodiester linkage needs to be destroyed, and that is carried out by a debranching enzyme, which converts this into a linear intron RNA molecule where it contains a 5' end and a 3' hydroxyl end, and this is the adenine at the branch point, and then there's some sequence in the middle. So this RNA molecule then can easily and quickly be degraded by the ribonucleases. And these SNPs which are dissociated, they can be reutilized in order to carry out another splicing reaction. So we are left with the spliced exons here. So this is how an intron is removed, and the two exons are spliced together by the spliceosomes. So I think that's sufficient for today. Thank you very much.